Okay, right. Thank you, Fran. I think we're going to have another talk from you now.
Yeah.
So hand back to Nikkon.
Thank you, Andrew. So Fran, you can switch out your screen. So the next talk Fran will be giving is a lightning talk, and that'll be followed by two pre-recorded talks. So I'll present those from my side, and then we'll take questions after the three lightning talks. So Fran, whenever you're ready, you can
Thank you.
switch over to the next one.
So the background for this talk is trying to look for chicken-associated Campylobacter types and how they may transmit around the industry. So we have a number of issues then to sort of think about. So firstly, the sampling regime. How many birds do we need to sample when we're talking about 20 to 50,000 birds in a shed? And how often do we need to sample? Because we know that things can happen very quickly. What's the best sample to take? Fecal samples or cecal samples? And then how many Campylobacter colonies do we need to follow up from a sample to assess the genetic diversity, both for individual birds and for flocks? And then also we have issues with culture. So Campylobacter is quite difficult to grow in the lab, and we know there's issues with it persisting in a viable non-culturable state. And then there's questions over the sensitivity of detection. So sometimes we can see DNA is present, but we're not able to grow the bacteria. We know that culture can lead to sort of differential selection of species or genotypes. And then also there's the question with young chicks. So again, there's groups in the US who can detect Campylobacter DNA in reproductive tracts and chicks in the hatchery, and yet it is possible to grow Campylobacter occasionally, but not very easily at all. And then particularly in this talk, I'm focusing on commercial flocks. So it's easier to, it is possible to grow Campylobacter from experimentally infected chicks quite easily, but I've only ever seen it on UK farms in commercial flocks once from a flock at 14 days and more typically from 3 weeks onwards. So as a pilot study for this piece of work, I did follow up a number of different colonies from an individual sample. So I looked at broilers at the bottom here, a broiler bird and a breeder bird. Obviously we repeated it a few times, but just showing you the results for one. So we do have to, for our samples, follow up in the region of 100 colonies per plate to start to see some variation in the different Campylobacter types. So obviously, if we want to sample lots of birds, picking 100 colonies per sample is just going to be completely impossible. So what we've developed instead is a kind of microbiome type study, but we're using a fragment of the poor A gene. So that is a gene that we've worked with for a very long time in our other typing schemes. And it means that we're sort of selecting more for Campylobacter. But then, yes, we've removed the need for culture and culture bias. We performed the sequencing on an Illumina MySeq and assembled the data with the Data2 pipeline. So I apologize, there's lots of stuff going on this slide, but this is a paper that was published last year. So if we start the top left panel here, looking at the results from the fecal samples, each bar is representing the poor A types that we recover from a single bird. So each different color is a different poor A type. And I've just sampled 5 birds per flock there. So at the top left-hand side, we can see flock A, B, and C are broiler flocks. So each of those birds, generally speaking, has a predominant poor A type of Campylobacter, and that would be repeated in the other birds in the same flock. And then on the right-hand side of that panel, the breeder birds, these are obviously much older, and as you might expect, they've got more diversity in the strains that we can see. And then if we look at the bottom of that panel, these are cecal content samples, so they generally are showing the same picture, but we often see a second co-dominant poor A type in the broiler birds. And then we expanded this work to include a lot more flocks from the UK, France, and then also a flock from Switzerland. So if we look at the culture positive broiler flocks first, so these were culture positive just before slaughter, we can see a similar picture where the individual birds are have a predominant poor type of Campylobacter, which is repeated, generally speaking, in the other birds. And this trend was upheld with Simpson's diversity analysis and Shannon's diversity indices and so on. So the culture positive broiler flocks have low diversity or dominant poor type. We also looked at the flocks that were culture negative, and actually we found poor DNA there, but more diverse. And then this we really weren't expecting. We also looked at, they would be different birds, but from the same flock between 1 and 3 days of age. And again, we were able to detect the poor Campylobacter DNA, but with greater diversity of poor types. So our working hypothesis is that we think that Campylobacter might be present in many young chicks with high diversity. And then there's something that predisposes certain flocks and a good number of flocks to then become overgrown with a predominant poor type Campylobacter and gut dysbiosis potentially during their rearing period. I'll finish there. Thank you for listening.
Thank you very much, Fran. So I will play the first, if you stop, there we go. I will play the first clip. And that is coming to us from Nikkon Sarker from the Child Health and Research Foundation in Bangladesh.
Hello everyone, this is Nikkon Sarker from Child Health Research Foundation, Bangladesh, and I am here to talk to you about Campylobacter in rural Bangladesh. It is well known that Campylobacter is a causative agent of diarrhea, and in Bangladesh, one study even showed Campylobacter infections to have a significantly negative impact on children's growth. However, there is still a lot we don't know about Campylobacter in Bangladesh, from its transmission pattern to its genomic diversity. This is due to insufficient sequencing data, and which is what we aim to change with our longitudinal study in Mirzapur, a rural area in Bangladesh. In our study, we are collecting human and various animal stool samples and doing both culture-based and whole genome sequencing technologies. Direct culture of stool samples on mcCDA media is done in our Mirzapur lab, and then the samples are transported to Dhaka for the molecular work, including DNA extraction, PCR, and sequencing. With the culture method, it takes almost 6 days to confirm Campylobacter, whereas with the molecular method, we can accomplish this in just 2 days. Till now, in our study, we have found 4.5% human samples and 18.3% animal samples to be positive for Campylobacter. In human samples, we found equal number of Campylobacter jejuni and Campylobacter coli, but this was not the case in animal samples, where we found 64% Campylobacter jejuni and 36% Campylobacter coli. We isolated Campylobacter from chicken, from duck, pigeon, cow, and dog stool samples. And now let's talk about the Campylobacter we found from our samples. I am very excited to share the sequence data which we have generated from our first ever Minion run for Campylobacter in Bangladesh. We have selected 7 isolates for sequencing from 2 different households. These were selected in particular because Campylobacter was found in both human and chicken samples collected from these households. All the 7 sequences have typical genome size of Campylobacter and 6 of them carrying the plasmids. The sequence data shows that 5 of the 7 isolates have the same antimicrobial resistance gene, and 3 of the plasmids carrying the tetracycline resistant TATC genes. The preliminary data from the phylogenetic analysis showed no diversity of sequence types or resistance gene profiles from multiple isolates within a single sample. From this dataset, long-read sequencing of Campylobacter in Bangladesh demonstrates genomic diversity in samples collected from the same household. And these are just the preliminary results from our study. There is much more to come as we culture and sequence more samples. Till now, the achievements from our study include successful completion of the first ever nanopore sequencing of Campylobacter in Bangladesh. We have also successfully isolated Campylobacter from human and various types of animal samples, and we identified sequence types of epidemiological relevance in Bangladesh. Finally, our hope is that at completion, this study will fill the gap in data and highlight the genomic diversity and the epidemiology of Campylobacter present in rural households in Bangladesh. There will be another talk about this study design and epidemiology later in this session. Okay, I would like to thank Bill and Melinda Gates Foundation, George Mason University, Quadram Institute, and all the authors and other people involved in this study. Thank you everyone for your time.
Wonderful. Thank you very much, Nikkon. And we'll go to the next speaker, who is Ann-Katrin Llarena from Norwegian University of Life Sciences.
Hi, my name is Ann-Katrin, Associate Professor in Food Safety at the University of Life Sciences. I'm going to talk to you about rapid detection methods of Campylobacter in chicken flocks. So we need those to identify positive the chicken flocks that are positive for Campylobacter as close to slaughter as possible, because a lot of these flocks, they turn positive during the last period of rearing. Today, we already have a surveillance program in Norway which tests chickens for the presence of Campylobacter approximately a week before slaughter. That is maybe a little bit too late, and that's due to logistical challenges. So our goal with this test was to see if there's any commercial method available that can rapidly and robustly identify Campylobacter positive flocks in a cost-efficient way. So we tested 4 different methods in a pilot and chose the best performing methods in a field study. We had 2 immuno-based methods, which are the Minividas Campylobacter assay and the single path Campylobacter. We also have 2 DNA amplification methods, the film array gastrointestinal panel, which is a multiplex PCR, and then we tested Campylobacter isothermal amplification assay on a Genie2 instrument. The test material was 10 pooled cecal swabs from each flock, and we compared the results in the rapid test with those achieved in a qPCR. After the pilot, the Campylobacter isothermal amplification assay was chosen as the rapid detection methods in the field study. The results of the pilot are given here. So in this bar chart, the percentage of negative samples that were also positive in the rapid tests are here in the bar charts as orange, and the positive samples that were also positive with the rapid tests are here in the blue. So basically, you would like these bars to be close to 100. And those that are the closest are the film array GI panel, which correctly identified over 90% of the positive samples. And around 70% of the positive samples were also identified correctly by the Campylobacter isothermal master mix on the Genie2. The 2 immuno-based methods performed not as good. However, due to the high cost of the film array and rather low throughput, it was the Campylobacter isothermal master mix which was chosen for the field study. So here it was the slaughterhouse workers that performed the analysis on the slaughterhouse on samples taken by the farmer and transported by the slaughter truck. So according to this test, in which we tested 122 samples, 65% of the positive samples by qPCR was also positive by the rapid method, which was the isothermal amplification. There's a little bit lower than the pilot, which might be due to the fact that it's now performed in a slaughterhouse by slaughterhouse workers. So the conclusion of the journal, which we probably could of the study, which we could probably send to Journal of Negative Results, is that even though the film array performs very good, it's too expensive to be rolled out at a large scale, and therefore we don't have any reliable method rapid of these 3 remaining ones. An additional finding was, however, that 40% of the flocks, or 38% of the flocks that were positive at slaughter, was actually negative a week before. So we do need better rapid detection methods. And with that, I leave you to it, and I acknowledge my funding sources and collaborators. Thank you.
Great. Thank you to all the speakers.
Thanks, Nicole. So I think we've got time for perhaps one question for each speaker in the interest of the time. So if we go in order, so we have a question for Fran from Ann-Katrin. So considering the rich diversity that you find, would you consider Campy as part of the core microbiome of the seeker of these birds? And also, given the rich diversity, that would indicate a number of introduction events. Were these free range?
Well, they weren't free range, actually. They were commercially housed. So I can't tell you where they're all coming from. And that's a really good question. But it does lead me to think that it probably is part of the core genome or microbiome, sorry. But I don't think that, based on other work, it's supposed to be there as a single type. I think its healthy state, if you like, is the low prevalence and more diverse, and not slightly higher prevalence and dominant. So yes, I think it probably is, but as we've seen in previous talk, there are sort of more issues coming up that we think that sometimes it can cause a problem for chickens, but I think that's in certain situations, and there might be some tweaks to management and so on that we can do. So that's my current way of thinking.
Thanks, Fran. Do we have Nikkon with us?
Yes, he should be there, but he may not be able to do video with broadband problems.
Nikkon, are you with us?
Yes, I am.
Hi, Nikkon. So firstly, a comment from Nabil, who says it's good to see the use of nanopore sequencing. And a question has come in asking how many isolates were taken per sample.
Okay, initially we are taking actually 2 to 3 isolates per sample.
Okay, thank you. There's a follow-up to us. Would you expect to see more diversity with more isolates? I guess the answer to that is probably yes.
Yes, we are going to do more because we have another 100 of samples isolates in our store. So we are going to sequence all of them, and hopefully we'll publish the data soon.
Okay, thank you. And lastly, for Ann-Katrin, so Ann, you talked about the week before the slaughter. So there have been a couple of questions on that. I'm going to change them around a bit because I think in your last question or your last comment, you sort of talked about it. So how rapid is rapid?
Within 2 hours.
Wow. Okay.
Yeah, because that's the time you're allowed to wait as a chicken. You're not allowed to wait anymore for slaughter because you need the answer. So you were aiming for that.
Brilliant. Okay, thank you. We'll move on, Nicole.